All right, so I'm Manuel Mameli, and so Christian and Garrett asked me to come over and give you a little overview on the dark side perspective of addiction. So as far as I got from this presentation, if I asked you, okay, what is the system that most represents and contributes to addiction, does anyone can comment on this? What is the most neuromodulator that plays much of a role in this context? Come on, bring it out. All right, dopamine. So basically, I came here to kind of stand out and say, like, yes, maybe, but it's not the only system we are going to look at. So how about we take a zoom out from this perspective and look up what else can be implicated and in which context. So needless to say that me talking for 2 hours and a half or 3, I'm going to be like drying out, probably die, if you don't intervene and ask a question. So please feel free to make it animated. So maybe you already touched base with this point. So we are in a dynamic life, we are exposed to external stimuli, and the majority of these external stimuli, hopefully for you, they will be probably positive and like reinforcing our behaviors, right? I mean, from food, music, love, sex, drugs. I don't know whether this applies or not, but we have the tendency to experience something and then to keep pursuing it because that is positive and we keep going. For me, like the most representative one is a pot of Nutella, and Christian knows about it. However, we are also like bombarded by a series of external stimuli that we experience as a negative situations, right? And so what do we decide to do? To pursue it or to not pursue it? Not pursue it. And if we are not pursuing, we are trying to avoid them, right? But then if we don't pursue them and we keep them like as a negative stimuli, and we learn about them, we anticipate our move such that next time any cue that would tell us about this negative stimuli, we're going to anticipate this and maybe not to do the action in order to avoid that eventual danger. And here you see a couple of representations like high stress, pain, fear, depending, strong vodka can be kind of aversive. But basically what it means is that we have the way of encoding rewarding stimuli and aversive stimuli and then adapt our next move to either pursue that positive or avoid that negative. Now, when you get into an aversive type of state, sure, you have a feeling of repugnance towards something, you have the desire to avoid, again, that experience, you don't want to do it anymore, or extinguish a behavior that is associated with that external stimuli. And so, indeed, what you have is that with a noxious stimuli or a stressful event, you try to get out of the situation. Now, you develop this coping strategy that is avoidance. Next time you're going to see like my name on the panel, you're going to say, like, maybe I'm going to switch presentation. I'm not going to go there. Now, you can have a different condition, which is you can have a single unexpected event, and then you're sure that you're going to have a coping strategy. But what if this negative event is repeated over and over? And what if you can't, in that condition, have a strategy or you are threatened by that continuous stimuli? So avoidance is not anymore a strategy. The event that is negative is repeated. And repeated over and over, it has been shown that this can get you in a condition that almost lead to disorders, to disease state. And actually, this is the base of at least these two disorders, mood disorders and addiction. Okay, and I'm going to try to tell you why this can contribute to addiction over this couple of hours. But basically, you get into a depressed mood or irritability. You get into diminished pleasure in activity, loss of energy and anxiety. So something that overall would say like, okay, I cannot get out from this negative situation. Now, in terms of circuit, I guess what you heard a lot during these days is the role of maybe dopamine neurons, the role of maybe GABA neurons in the midbrain, how this microcircuit is controlled and acts over drug intake. But then, little question for you is, are these dopamine neurons like able and capable to have their own reasoning? So they don't need anything else, and they just act controlling our behaviors. My point of view is that this dopamine neuron, they need maybe someone or many players that at a moment or the other would say, well, okay, now is the time to encode this. Now is the time to encode that, because you are in a different condition and situation. So in the lab, we try to look at what is that can modulate the midbrain dopamine system in this condition, and especially in this avoidance aversive encoding. So the talk will run around the role of these structures called the lateral habenula that is located just in the middle of the central nervous system here, just underneath the dentate gyrus and the far end of the thalamus. And I'm going to try to convince you that there are situations in which this region can play a role in specific encoding, and that this encoding is actually may take part during addictive or be complementing addictive behaviors. Now, I'm Italian, and like you cannot start anything not thinking about food. So the lineup of the day is basically taking you through a couple of specialty of the region, which are the piadina, the tortellini, the galletto arrosto, and the migliaccio. Which basically is going to touch base with a couple of information, which are a little bit of the anatomy of the lateral habenula. Where is it, what it does, what is encoding. To try to give you some of the causal links between lateral habenula function and aversive states. And then to drive you through a little bit of the cellular, but especially the cellular and synaptic profile, but mostly tell you a little bit about the work we are doing in terms of lateral habenula dysfunction and depressive states, but in the context of addiction. So, if you have to go into the piadina menu in the functional anatomy, so I told you that this structure is located here, but this is a structure that has been like preserved during evolution. You find it in the zebrafish, you find it in the mouse, you find it in the humans. And although small, there are potential applications, even in humans, to selectively activate or shut down the activity of this small nucleus. Now, what's in the lateral habenula? What's in the LHB? Yesterday we were discussing with Garrett, and most of the papers describe this nucleus as homogeneously composed by glutamatergic neurons. So most of the paper out there state that the axons going out from the lateral habenula, which go downstream to the midbrain and then diverge to the raphe, they mostly release glutamate. Nowadays, there is still this little question mark on whether or not this is the only phenotype that we have in the LHB. But for the moment, it's considered a kind of glutamatergic, although there are reports of several neuropeptides that are present and expressed in this structure. But for the sake of simplicity, think in this order. Glutamatergic neurons in the lateral habenula projects glutamatergic axons down to the midbrain and down to the raphe. So These neurons also receives several inputs. And therefore, all these inputs would, if they are glutamatergic, would activate glutamatergic neurons in the lateral habenula, and then would control the midbrain dopamine system. Now, what are the inputs first that can control this structure? There are several evidence out there telling us that seemingly there is a glutamatergic input from the cortex to the lateral habenula, mainly in rats, rather. That these neurons receive basal ganglia inputs that are both glutamatergic and GABAergic. And if you want to retain something about this, well, retain this. Globus pallidus internal capsule, so a basal ganglia structure, an output structure of the basal ganglia, can project terminals to the lateral habenula that co-release GABA and glutamate. Just stress this point because this is going to be taken up later on. Then recently Garrett has shown that the lateral hypothalamus mainly project glutamatergic inputs onto lateral habenula neurons. And we have also evidence that the VTA can provide a glutamatergic source and an inhibitory GABAergic source. So as you can see here, we have a structure that is in the middle of the brain, funneling information down to the midbrain, so potentially able to control dopaminergic activity, and I didn't tell you yet how. And that this is under the control of a variety of structures that may encode multiple signals. Yes?
These are unidirectional, or these are unidirectional connections or bidirectional connections?
So that's a good question. So for certain of those inputs are a single direction. So the globus pallidus projects but does not receive from the lateral habenula. On the other hand, you raise a good example because this connection here, where neurons in the lateral habenula project to the midbrain, they also receive from the midbrain. So there is a feedback loop.
Yeah, are there any specific parts of the cortex which have particularly strong influence into the habenula compared to others?
I'll give you my hint. So my personal experience in terms of what we've done in the lab, we can't get this working very well in the mouse. So we don't see much of cortical afference. There are reports, although in literature, using retrograde tracing, where they see handful of neurons in the prefrontal cortex that light up with this retrograde tracing. Now, this is still debated and has to be clarified. So it's still missing what is the topographical organization in cortical regions for innervating the lateral habenula.
So for the GPI, this is part of the direct pathway of the dorsal striatum, and so is the lateral habenula, how would it be connected to more ventral parts like the
It's a good question. So the problem is the following. We tried to see whether there was some connectivity between the ventral striatum and the lateral habenula, and seemingly there are axons that innervate the lateral habenula when we inject any anterograde down in the ventral striatum. What we discovered, unfortunately, is that the injection site was spreading in the ventral pallidum.
In the ventral pallidum?
Yeah. So we are not sure whether...
That could be indirect.
So, exactly. Yeah, let's put it like that. That when you use retrograde tracing in the LHB, you don't see much in the nucleus accumbens or ventral striatum. So there might be indirect ways of controlling it.
Because, I mean, yeah, so the VP would be the analogy of the GP, right?
Right. But we don't find anything in terms of a direct connection. There are, I think, a paper coming up from Scott Russo saying and telling us that probably D2 neuron, I think, they can send axons to the lateral habenula. But
D2 neurons
of the ventral striatum.
Oh, directly.
Directly, but
so without a relay.
Without a relay to be seen. Now, where do these information are funneled? I told you that these axons from the structure over here, they go down here to the midbrain and also to the raphe nucleus. Now, for sake of simplicity, today we are going to only focus on this part. How do lateral habenula neurons that send the glutamatergic terminals control midbrain dopamine neurons? There are two pathways that have been described. One indirect pathway that seems to be the major pathway, meaning that the terminals onto the midbrain synapse mainly on GABA neurons, and these GABA neurons then provide inhibition onto dopamine neurons. Then a smaller, somehow weaker pathway has been also identified, which directly goes from the lateral habenula to dopamine neurons in the midbrain. Now, question for you will be, this is the predominant pathway and connectivity. What will be the net effect onto dopamine neuron by activating the lateral habenula neuron activity? Inhibition. However, this is there. So that tells that somehow there could be some balance, and that either a single dopamine neuron is getting inhibited by a desynaptic circuit, as well as excited, or alternatively, two distinct populations of dopamine neuron may get either a desynaptic inhibition or a direct, and the other a direct excitation. Now, this is what I just tell you. Now, if this is the circuit, what is the functional evidence that these nuclei can indeed encode aversive states? But now, before getting in there, if you were to think out of the blue and say, well, this structure is encoding aversive situation, an aversive signal, through which pathway it will do so? It will do it through the desynaptic indirect pathway, or it will do it through the direct pathway, considering the broad knowledge you have on dopamine function. Say that again? Indirect. So your colleague here says, if the LHB encodes an aversive stimuli, it is because it excites GABA neurons in the midbrain and then inhibits dopamine neurons that are close by. Everyone agrees on that? Fine.
It can do it both ways.
Say that again?
It can be doing it both ways.
It could be doing it both ways. Can you develop it?
They're going to inhibit the gamma neurons to inhibit dopamine, but also excites dopamine, and it's different connection, different neurons, and they might be responsible for something.
So you are telling me right now that what Christian has said earlier, which is dopamine encoding, like eventually reward, and now dopamine, they encode, dopamine neuron encode aversion? No, yes, stand up, say yes. Come on. Right, so this is what you brought is actually correct. So not that Christian was wrong, far away to say that. But what you said is correct, and we are going to see later on. Indeed, you can have this aversion encoding via either the desynaptic, but the same aversion encoding, you can have it through direct dopamine neuron activation. But then, this you have to keep it in mind, because for the moment, maybe what you've seen is that activity of dopamine neuron would encode for, say, reward, reinforcement, and so forth. So that must be that there is some properties of dopamine neuron that are not all alike, maybe, that would encode different stimuli.
Would it only encode the anticipatory? If I'm expecting a punishment,
yeah,
and I don't get it, I mean, would that be part of that other dopaminergic
2 slides, and we get to the point. So what is historically, how researchers kind of got to know about this region first, and how did they get to know what it was encoding? The initial seminal work was done in monkeys. Basically, I guess you heard about the work from Wolfram Schultz in the encoding of prediction error of reward in monkeys and in dopamine neuron. Well, there is a sort of a mirror scenario in the lateral habenula. And so this is the work from Okide Kosaka at NIH, where they use as well monkeys, and they put them basically in the same kind of condition, and the task was basically a direction of their eyes towards, okay, this is the task of getting a reward, otherwise if you move there, you get no reward, okay? So the kids, they took this movie and they gave me the reward because I got all the tasks correct in the end. So basically, what did they do here? Monkey was doing the task. They were at the same time recording a single unit activity of these lateral habenula neurons while the monkey was performing the task. What you can see here is basically lines that reflect the activity of these lateral habenula neurons. So here, move eyes, there is no reward, there is a reward. Excitation of these neurons during no reward task, no reward phase, inhibition of these neurons during the reward phase. What they did in the same time was to have recording electrodes down to the midbrain, recording putative dopamine neurons. What do they see? Well, they see what Wolfram Schultz have seen. Basically, reward will drive the activity of these neurons. No reward would drive the activity, would decrease the activity of dopamine neurons. So there is a total mirror condition here between LHB activity and dopamine neuron activity when in the same task, one will be opposite of the other. One thing that they analyzed a little more in detail is that this guy here was firing slightly before the dopamine neuron. So time-wise, first the LHB activity was coming, then the dopamine neuron activity was coming. So this maintains this idea of a loop of lateral habenula neuron activity may be controlling downstream midbrain circuit and midbrain activity. If this is the case, what is the circuit that is mediating this type of responses? What would you say it is? The synaptic circuit or the monosynaptic circuit?
The monosynaptic, the quicker transmission.
Glutamatergic neuron or GABAergic neuron, this one in the lateral habenula? Glutamatergic, so they excite. So if they are excited here, the neuron is inhibited here, the dopamine neuron. If the lateral habenula neuron is inhibited, here you have an excitation. So the two things are opposite. So your colleague there was pointing out, okay, it might be two, but still the desynaptic seems to rule. So in other words, what it means is that the activity here seems to down-regulate the activity of dopamine neuron when a reward comes, and on the opposite manner, well, actually, when an aversive encoding or a no-reward situation comes, and the opposite happens when a reward comes. Here inhibited, and here excited. Here excited, and here inhibited. And this may occur through this desynaptic pathway, through these GABAergic neurons that are located in a sort of a next region to the VTA, which is called the rostromedial tegmental nucleus, but let's keep this as a pure anatomical information. Now, lateral habenula is a source of negative reward signals into the dopamine neurons. But so, is this activity then linked to motivated negative states? So what they showed us in monkeys for the moment is a correlation of if the monkey get a no reward or a punishment, there is a differential firing activity between LHB and midbrain. Now, is this behaviorally relevant? The question is. So here you can move all kind of critique you want, because the author of these papers is far away. I mean, it won't hear you. So be critic. I mean, raise, it's all published anyway, so raise up all the potential critique and questions you have. So what the authors here have done, basically, they try to really causally link the function of these terminals from the lateral habenula down to the midbrain. So they check a couple of things. They checked whether there is a behavioral relevant condition, whether the connectivity reflects either this desynaptic circuit or the monosynaptic circuit onto the midbrain. So circuit dissection and functional role at the behavioral level. So tell me straight, what do you see here? What is this and what this picture represents? Just describe, what is this? What is this? You see here D, V, M, L. You see here instead D, A, P, V. What do you see here? See? Dorsoventral. Yeah, okay, but this picture, what is it? Oh, coronal. Someone said coronal. Okay, coronal. So this is a coronal plate, a slice in a coronal plate. What is this instead? That's a sagittal plate. Okay, so we have a picture in the coronal plate and one of the sagittal plate. Is going to be the same animal? Okay, no. Okay. Now, what do you see here? This is an injection of like adeno-associated virus coding for chynodopsin, as you can see here. And you see all these somata that are labeled by the virus. What do you see here? If these guys are green, are injected here, what do you see in the picture? Okay, so a good labeling in the CA1. Okay. Now, what do we see here instead? So this is, so the injection, let's say the injection site is here, and here it's a zoom in into the fibers that I told you they funnel down to the midbrain. So you see all these axons that goes down to the midbrain, and you see they go up here, but you see quite a bit of green here on the back. So they are innervating nicely these midbrain areas. So that's anatomical, and anatomically confirm, like the cartoon I showed you earlier, there is these glutamatergic axons, they run down to the midbrain, and they are present here. Now, are they functional? So what the authors did was to prepare slices of this midbrain, shine light in order to activate these terminals, right? And to record then responses into potentially GABA neurons and dopamine neurons, right? And so what they looked at was what is the connectivity rate, what is the likelihood of getting a response in these different populations. So I guess it's all fine with everyone. TH negative stands for? Say that again? Tyrosine hydroxylase negative, meaning? Okay. Non-dopaminergic, but sitting in the VTA. These ones are dopaminergic, sitting in the VTA. And these are rather instead GABA neurons located very posterior to the VTA. What you see here, basically, is that the light-responsive neurons, here in the posterior VTA, there was a lot of this GABA neuron responding to light illumination, to the stimuli of these lateral habenula neuron terminals. What you can see here is that when you are in the VTA and you record this TH negative, so non-dopaminergic, presumably a lot of GABAergic, but potentially some glutamatergic, then you still get a good 50% of responses here. However, when you go and you record TH positive, putative, dopaminergic neurons, only 5% present response. So, what does this mean? Going back to the conversation that we had earlier.
Lateral neurons are not projecting to TH positive neurons. It's
Right, okay. So put it on the positive sentence. So lateral habenula neuron projects where?
To GABAergic.
Say. Okay, so seemingly this desynaptic inhibitory pathway seems to be more prominent than the direct pathway. So, equal, the prediction will be that the lateral habenula neuron would dampen activity of dopamine neuron, all right? So, would you ask that to the authors? If you were a very objective, nasty reviewer, would you ask this information to the authors? To know what happens to the dopamine neurons, if this is the truth?
Not necessarily, right?
Not necessarily?
Because it's not projecting to dopaminergic neurons, so it could not even have an effect on them. It might because it's probably inhibiting aversion. But
right. But I mean, one thing we were discussing here is whether these motivational states, either rewarding or aversive, would be actually as a readout of modulation of dopamine neuron activity. Now, this is probably the most prominent pathway. Now, what they did was to go into in vivo modulation of this pathway, insert a fiber optic able to stimulate these terminals in the midbrain and mostly, therefore, activate this desynaptic pathway. And I don't know whether, it's playing. So this is from the website, and this is, of course, work from Garrett. And this is basically like the first demonstration. And so this is only the control. The mouse shuttles from one side to the other. Let's say that is the first demonstration in vivo, providing information at the circuit level and at the cellular level that LHB neurons can modulate midbrain function and then drive to an aversive behavior. Every time the animal crosses and puts his head over here, lights shined and the mouse turns and goes into the other side. So the mouse, what it does basically is avoid totally the compartment submitted to stimulation. So when you stimulate these glutamatergic terminals onto the midbrain, then you have a complete aversive state overall. Not only that, but there is an important aspect that is, to me, crucial. If you start pairing or conditioning the mouse to one compartment with the light, over several sessions, you put the mouse in, you don't give any light whatsoever, you let the mouse free to choose one or the other compartment, the mouse will stay away from the light-stimulated compartment. Now, what does it mean? Anyone can kind of develop what I just said?
You develop synaptic plasticity to generate a learning response that's completely post-aversion to the place that's...
Whether we develop a synaptic plasticity, let's say, potential, not given, but at the behavioral level, there is, I think you brought up the good term, there is learning. Correct. So, stimulating these axons is sufficient to generate a form of learning that place, no good. I'll stay here. So, not only these axons generate a place aversion, but they can engage a memory trace. So, take-home message basically is LHB neurons seems to be mainly glutamatergic. They project glutamatergic terminals to the midbrain. They seem to connect mainly to GABAergic neurons in the midbrain, potentially inhibiting dopamine neurons in the midbrain, and they can produce passive and conditioned behavioral avoidance. Okay? So first demonstration that there is a functional role in aversive encoding. Still, we discussed like 10 minutes ago that there is, although a weaker, there is a direct pathway here. And this direct pathway seems not to go to GABA neurons, but seems to go directly to dopamine neurons. So, same here, another paper comes out, and what the authors have done in this paper was basically the following. They used a rabies virus-based strategy. So the RV was also encoding for diopsin, channelrhodopsin. So the RV has the property that jumps a synapse and goes retrogradely. Okay, so it would infect lateral habenula neurons that would project to the VTA. So what the authors did here was to stimulate the somata of these neurons located in the LHB that they were aware were projecting to the VTA. Again, stimulating these neurons, what they find is the same thing that Garrett and his group has shown. There is an aversion and also a conditioning that takes place when shining light on one compartment. Behaviorally speaking, same results. Now, what they find, however, was a slightly precise information, which is these are dopamine neurons that projects to the medial prefrontal cortex. So you are left with the idea, terminals from the LHB mainly contact GABAergic neurons and a handful of dopaminergic neurons. What they find here is that some dopamine neurons, and precisely those that project to the medial prefrontal cortex, but not those projecting to the lateral shell, they respond to light. So, terminal from the LHB, they can innervate a population, a fraction of dopamine neurons in the midbrain and GABAergic neurons in the midbrain. But these dopaminergic neurons seems to only be those going to the medial prefrontal cortex. They find as well that GABAergic neurons in this RMTG, the same one that I described you previously, they also respond, so this was good. But they also find some dopamine neuron with a desynaptic GABAergic response, although few. So, bottom line for this finding is that it seems that an indirect pathway is ruling this connection, but then the weak pathway, these dopamine neurons going to medial prefrontal cortex also receive these lateral habenula neurons. Well, fun enough, these dopamine neurons going to the medial prefrontal cortex have been described as dopamine neurons that encode aversive stimuli rather than rewarding stimuli. So, another take-home message is dopamine neurons seems to be not all alike. They can serve probably opposite function depending on their target projection, and probably depending also on the type of input that they receive. Dopamine neurons projecting to the NAC lateral shell are those that have been described to mainly encode rewarding signals. And indeed, they do not receive inputs from the lateral habenula. So, in conclusion, there are these two pathways that overall, through distinct circuits, they can encode aversive stimuli. Now, there is one thing that in my mind is still missing. And do you have any questions after this portion?
Yeah. It's interesting, so this 5% of hypothalamus habenula neurons in the VTA also project to the cortex. Yeah. Did you see anything or that they reported where in the habenula? So is it like lateral, sort of specific population in the habenula that projects to the VTA?
Not for the moment. not that I know, there is no topographical organization in the lateral habenula of neurons that projects to either the GABA or the dopamine neurons. This has not been described. And I think you raised one good comment that I think is still missing and we don't have information on, is those neurons that receive the desynaptic connection from the GABA neurons, where do they go? We only have a hint that they may go maybe to the ventral striatum, but we are not sure about it. We definitely know that these neurons receiving the direct connection go to the medial prefrontal cortex. Any other question? Yeah.
Yeah. Aversive stimulus are known to be consolidated. That is, after the experience, the person may likely have cues of aversive stimulus. In this model, how does the organism consolidate the aversive stimulus for subsequent memory utilization in the next cell?
So, the work that has been done in monkeys also suggests the following, that when the animal perceives an aversive stimulus, you then can condition the animal and associate a cue with that aversive stimulus. Then there is a shift of the activity of the lateral habenula neurons from the aversive stimulus to the cue. So in the LHB, there might be forms of plasticity learning and acquisition that shifts the activity of neurons from the stimulus to the cue, as it happens in dopamine neurons. Now, your question goes beyond that. How this, say, cue and this association is encoded downstream, not known. We know that this lateral habenula neuron can do it, but we don't know how these, say, learning processes are translated from this above circuit or structure down to the downstream target.
So if those dopamine neurons that project to the PFC are aversion dopamine neurons, right, then you would predict that if you were to activate them selectively, you could do your dynamic CPP or induce any form of aversion.
So
has that experiment been done?
That experiment has not been done. Can be done. Yeah, you can do it. No, you can do it. We could do it, but I mean, it's so possible to be done.
Why haven't we done it?
Why?
Yeah.
Why?
As a field.
There was a paper that just came out, and I didn't read it yet. Maybe someone in the audience has. That at least in the abstract, maybe it shows or claims that projection is more important for overall salience and doesn't have a valence.
So which paper is that?
It was just published in PNAS. I'm afraid I'm.
Which one?
Which one?
There was a Nature Neuroscience paper and then a different one.
Yeah.
Which one?
I'll find it. I don't remember the author. So.
Because and they stimulate only the dopamine to prefrontal cortex.
That's my recollection, but.
Okay, because the only thing is known is like a little plasticity experiments that has been done, where if you were to expose the mouse to very like aversive stimuli, you do not have a plasticity in those neurons, those dopamine neurons projecting to the ventral striatum, and you only have a certain plasticity only in dopamine neuron projecting to the cortex. So this also,
this has been done, right?
Yeah, this has been done, but the specific modulation of those neurons, I'm missing.
So what's the difficulty? Why is it a little bit more difficult than?
To me is, so what people has described is if you were to inject retrograde tracers in the prefrontal cortex, the amount of dopamine neuron that you see labeled is apparently really, really small. So is this a technical difficulty that we can overcome? Garrett, I don't know whether you know anything about it.
So how would you do it if you have to do it?
In terms of just selectively?
So because what you want to have is projection and cell type specificity.
I mean, you could try the terminal stimulation experiment, right?
Yeah, but again, the difficulty is really the divergence of the projection, which to the PFC is huge. So even with the terminal stimulation, how many terminals are you
It's
going
too
to
sparse.
eliminate?
Marina?
It was done pharmacologically many years ago. I feel like such an old person. But in the early to mid-'80s, Bob Roslav took advantage of the fact that the mesoprefrontal neurons are selectively sensitive to anxiogenic beta-carbolines, and so they could drive them that way, and that was associated with stress responses. And I think that they showed using extracellular single unit recording that they were selectively activating mesoprefrontal neurons, not some of the other populations. And definitely it was anxiogenic. So I mean, that's not the proof you're looking for, but it certainly helps. You know, a bit.
Let's say that, go ahead.
I had a big picture question. Given that what we know about the amygdala with your diversion, how does it fit with the habenula? I mean, do we know if there's a direct connection? Or is it possible now when we're talking about this that is it a more downstream player through the prefrontal cortex connecting down to, maybe that's the kind of loop.
And the third point I would say is necessary that there is a loop, or that these are parallel circuits encoding slightly different properties, one emotional and the other through a basal ganglia, more motor-driven, escape type of behavior. So this is a big picture that people is we are still trying to understand. But one thing we know is that from the amygdala directly per se, there is no connection with the habenula. So they might converge, although at one point or the other, say the midbrain, actually.
So when you can generate an adverse response by stimulating the LHB, but if you just stimulate an adverse response through a foot shock, can you ablate that adverse response by, ablating the LHB, or do you, if you just do the adverse response, do you see an activation of the LHB with CFOS?
Yeah. So people have done it. Paul Shepard has done it with CFOS. You see a CFOS activation when the animal is put into like an aversive condition. In the lab, we have evidence that also in mice, when you have the animal even anesthetized and you record the single units in vivo, then that foot shock increases the activity very physically of these lateral habenula neurons. So there is a direct response. There is an encoding there. Then what is the loop? What is the circuit based of this response? This is something we are looking into that, but not clear so far. Maybe one thing going back on how can we identify these dopamine neurons projecting to the cortex and then being able to, say, control them. Maybe one way will be to use this immediate early gene expression such that we can drive, say, protein expression driven by the activation of this immediate early gene. So if these were the only one responding to the shock, can we drive, I don't know, ARCs, FOS, only in these neurons, only these activated neurons, and then lead to the expression of, say, chynodopsin or whatever opsin to control this population? But that has not been done yet. Questions? No.
Just one last question.
Yeah.
What happens to the rodents if you take out their lateral habenula behavior?
So gross behavior, not really much. So they can cope really well with locomotion.
So what do you mean take out?
I guess like lesioning.
Yeah.
So in terms of lesioning, if you lesion the lateral habenula, like gross behavior are not affected. Like no weight loss, locomotor activity seems okay, let's say, and I think they measure also food intake. But then if you put them in context of challenging the rodent, say, self-administration of sucrose, they don't perform. Self-administration,
They don't learn to
they
self-administer?
they learn to self-administer, but then they decay very fast. Then,
but they do learn to do.
But they do learn. They do learn to self-administer. I think this is not done. I think
And what
this is
happens
not
if
done.
you stimulate the lateral venula?
Okay, that's another thing that is not is not published. We did some pilot, and I know another lab did also pilot in that, like simply like a fiber optic stimulating lateral habenula neuron plane. I don't know
Like one
if you
would
guys
do
done.
with deep brain stimulation.
The animal sits there, and that's it. You overactivate these neurons at several frequencies from 20 Hertz to up to 40, and a normal locomotor activity stops there, and that's it.
Like a catatonia, that thing?
I don't know whether it's catatonia, it just stops. Now, maybe we can
we
make a
can
link
ask you.
when we start talking about dysfunction of these nuclei.
And do you know, I know that people are attempting to do deep brain stimulation in
They are
venula
doing it rather
in
than
humans.
Yeah, they are doing it rather
And
than
yeah, I
tempting.
know at
Yeah.
Sinai, you know, I know that they're doing it.
In Germany, like there is a group
Germany
actively
probably
doing it.
Are there any reports on behavior, on how, what people feel or what they report?
There is so
When
the
it is
report from the guys in Germany says that the overall feeling of the deep brain stimulation is rather positive. But who were these people? These people were were depressed people.
I mean, it's one case.
They have more now. They have more now. They have an entire cohort. So they claim they can ameliorate the depressive symptoms in these patients. This is, I mean, few patients that have been implanted,
Yeah, there's
but
freaks.
that's.
I know some people will do that, and they're trying to do it now in addiction.
This
so,
I'm not aware of, but okay.
Yeah, it's
Okay. Okay.
To find a good.
Yeah, maybe
subject.
okay. Then what we will see later maybe will be useful to explain why using this approach in the context of addiction. Okay, so we've seen now basically a little circuit where this lateral habenula controls the midbrain. We've seen how these terminals can be behaviorally relevant. Now, I'm a cellular physiologist, and I wanted to go down into what is generating activity in these neurons. So what are these neurons? So these neurons are kind of sparse in the structure. They have rather a non-complex dendritic arborization, but they still have spines where potentially they are receiving glutamatergic inputs from. What do, at the level of the molecular traits of these neurons, where they have spines and they have receptors that can sense glutamate, therefore NMDA, and AMPA receptors. And these AMPA receptors are kind of peculiar. I guess maybe you might have heard it from Marina and Christian. There are different types of AMPA receptors, but mainly two types in terms of calcium permeability, non-calcium permeable and calcium permeable. Well, if you heard about plasticity in the VTA or in the nucleus accumbens with the insertion of calcium permeable AMPA receptors, in the case of the lateral habenula, these calcium permeable AMPA receptors are there at baseline. So they are present throughout development actually and adulthood. Then they present GABA synapses, GABA A receptors with different subunits. We know that they possess GABA B receptors and they possess mGluR1 receptors, and in small amount, mGluR5. So those is what they are constituted of. Now, while we were talking earlier about deep brain stimulation in this area and depression in this area, because several years ago, there has been a report suggesting that by implanting humans, actually depressed patients, with deep brain stimulating electrode in the lateral habenula was able to kind of remit this patient from depression. This has been pursued, and then over time there have been a couple of instances showing that in animal models trying to modeling mood disorders indeed, the same thing would apply. So you could rescue a depressive-like phenotype by acting on the lateral habenula. Why is this? So now we have a region that encodes aversion, and it's a region that is actually targeted for mood disorders. Why targeting this region for mood disorders, first? Second, what is supposed to do the deep brain stimulation in this region such that it would reverse the symptoms of a depressed patient? So what is it that is doing to the physiology of those neurons? Well, back in time, about 15 years ago, what it has been found but did not attract much attention was that in depressed patients there was a sort of a, they describe it as a calcification of the habenula in humans. Then at one point there was another study suggesting that the metabolic activity in humans was highly increased in depressed patients. So 10 years after, these papers were kind of retaken in consideration, and the hypothesis that was drawn was, well, in mood disorders, in depressed patients, it seems to be that there is a hyperactivation of this lateral habenula. And the conclusion after the deep brain stimulation approach was the deep brain stimulation is shutting down the activity of this structure and then allowing the patient to be remitted from his phenotype, from his depressive-like state. But at that moment, there was no functional basis of it. All right? So this kind of raised the interest onto this structure. And just to give you like a little into it, when you look at mood disorders, you are looking at patients with low mood, behavioral despair, anhedonia, or loss of motivation. And you see here loss of motivation. When we talk about motivation, we think about midbrain. So there is anything to do in terms of lateral habenula dysfunction, midbrain function, and addiction in this case. So this is what I would like to draw you through. So this is actually this initial paper where they implanted the DBS electrode and they remitted this patient from major disorder. But so this is one of these papers indicating that there is a certain activation of these nuclei in depressed patients. But what a couple of papers suggest is that if this is a control slice, the chronic stress seems to produce an increase in C-FOS labeling in chronic stress mice. Not surprising, because chronic stress has been shown to be a model for depression. But as well, withdrawal from amphetamine seems to drive a very strong increase in C-FOS activity. And this is actually, this is acute and this is in withdrawal. So this was the first hint, the sole paper back in time suggesting, okay, there might be a link with activation of this structure, not only in chronic stress and stressful situation, and therefore aversive, but also in drug withdrawal. Why drug withdrawal? What did you hear so far about drug intake? We heard about, I guess, reinforcing properties of the drug, about a drug being able to increase the activity of dopamine neurons, so most likely driving what? Positive feelings. But then, is this the only phase of addiction? I mean, we talked about compulsion. There are theories out there saying that actually there is an equilibrium between the positive properties that the drug can have and some negative symptoms and negative properties that the drug can engage. And this, especially during drug withdrawal. George Koob has been one of the leaders in on this topic, basically indicating that there are withdrawal negative effects, and that these withdrawal are actually this withdrawal period is actually taking a large part in setting the negative and social and the personal consequences. So there is the compulsion, but why the compulsion is generated, according to this theory, was because of a equilibrium between I want to take the drug again because of the compulsion, but also why I want to take the drug again. And according to this theory is because I want to relieve these negative symptoms. This is, according to the DSM-4, the major points classifying major depressive disorders. Depressed mood and irritability, pleasure in daily activity, increase or decrease in appetite, psychomotor agitation or retardation, fatigue, loss of energy. Same DSM-4 says that psychostimulants withdrawal, they present several depressed mood and or irritability, loss of pleasure in daily activity, increase in appetite, psychomotor retardation, fatigue, loss of energy. So there are a couple of points that put in parallel major depressive disorders and some of the effects that arise and emerge during drug withdrawal. I think here we can take a break. Good.